Deoxyribonucleic acid, or DNA, contains the instructions for life. Many of our traits are determined by those instructions, like hair color, eye color, and blood type. DNA is made up of long strings of molecules called nucleotides. Each nucleotide is composed of 3 parts: a phosphate ion, a 5-carbon sugar called deoxyribose, and a nitrogenous base. It is the nitrogenous base that is used by the DNA molecule to store information. All forms of life use the same 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. DNA is a double helix with 2 sets of nitrogenous bases aligning with each other. These bases form weak hydrogen bonds with each other, and we refer to them as base pairs. Adenine always pairs with thymine, and cytosine always pairs with guanine. The sugar-phosphate backbone causes the 2 strands of DNA to twist around each other, forming a double helix. The 2 strands of DNA run in opposite directions. A section of the base sequence that codes for a protein is called a gene. A copy of the gene is called an allele. Every cell of every organism on the planet contains DNA. The only difference between you and a tomato is the length of the DNA strands and the specific sequence of bases on that DNA. In fact, the tomato plant contains more DNA and 7,000 more genes than you do. In prokaryotes, DNA forms circle-shaped segments of DNA called plasmids. In eukaryotes, DNA forms complex, tightly packed and wound structures called chromosomes. While most of the DNA in your cells is in the nuclei, your mitochondria does contain a small amount of its own unique DNA. When a cell undergoes either mitosis or meiosis, the first step in the process is the replication of DNA. Replication involves a special protein called helicase that unwinds and opens up the DNA double helix. Another enzyme called DNA polymerase then binds to the unpaired strands and creates a copy of the matching DNA sequence. The DNA polymerase does this by pairing up the complementary bases. So if the original strand says ATG, the polymerase will match it up with TAC. This results in 2 identical strands of DNA, each containing half of the original DNA strand.